Once the DNA it is immobilized on a solid spot, then it can be analyzed or studied by using a technique which is called as Southern blotting. So Southern blotting, it is a laboratory method that is used to detect the DNA fragment among a mixture of DNA molecules. As I told you earlier that this technique was developed by Edward Southern in 1975, so that's why the name of the technique it is after its inventor, so that's why it is called as Southern blotting. To perform Southern blotting, gel pre-treatment is required. Gel pre-treatment क्यों required होती है? Once DNA fragment has been separated, तो इसकी रीज़न ये है कि जब फॉर एग्ज़ाम्पल कोई genomic DNA का source है and it has been digested by using restriction enzymes, तो कुछ fragments बड़े साइज़ की होंगे, कुछ छोटे साइज़ की होंगे, and if they have been separated on the gel, तो कुछ fragments जो हैं वो slowly move करेंगे larger size की वजह से और कुछ rapidly move करेंगे smaller size की वजह से. So before immobilization on the solid spot, तो इसकी pre-treatment की जाती है, like depurination की जाती है, जहाँ जहाँ purine bases हैं उनको remove किया जाता है by the treatment with HCl or even alkali like sodium hydroxide can be used. तो जहाँ जहाँ प्यूरिन बेस होगी वहाँ वहाँ वो रिमूव होगी और डीएनए की जो लेंथ है वो शॉर्ट हो जाएगी और एट द सेम टाइम एल्कली जो है वो डीएनए को डीनेचर भी करेगी. डीनेचुरेशन मीन्स दैट द डीएनए विल बी कन्वर्टेड इनटू सिंगल स्ट्रैंडेड फॉर्म एंड इन सिंगल स्ट्रैंडेड फॉर्म नाउ दे आर रेडी फॉर हाइब्रिडाइजेशन प्रोसेस बाय यूजिंग a radio labeled probe. After transferring to the nitrocellulose membrane or nylon membrane, then fixation process it is carried out. Fixation means that the DNA it is strongly binded with the membrane and it is fixed so that it will not move after repeating washing. And although different techniques can be used, but baking it is the most commonly used technique. Once it is immobilized, then the nitrocellulose membrane with bounded nucleic acid, it will be placed in a solution of probes, and if complementarity is there between the bounded nucleic acid and the probe, then they will hybridize. When they will hybridize, then the hybridization process it can be visualized by autoradiography as I explained earlier. So this is the flow diagram of Southern blotting. Here in first step, the DNA fragments they are separated by agarose gel electrophoresis, and once they are separated, then they are transferred on the membrane, either nitrocellulose membrane or nylon membrane, and then they are fixed. After fixation, then the nitrocellulose membrane it can be placed in a solution of radio-labeled probe here in the third step, and where the complementarity it is there, that if it is probe having the complementary sequence to the target DNA, then a base pairing it is there, and then at this step we wash the membrane so that the extra or unlabeled radioactivity will be removed, and in the fourth step the membrane can be analyzed by autoradiography by using X-ray film. So this is another diagram indicating the process of Southern blot hybridization. So here it is the source DNA, and then it is restricted by using restriction enzymes so that the different fragments are there, and then they are separated by gel electrophoresis here. And after separation, here the gel pre-treatment it is required, as I mentioned, depurination treatment, because after restriction analysis different type of fragments they may be generated that will be separated on the gel, and then it will be transferred on the membrane in the next step. So this is the membrane, nitrocellulose membrane, where the DNA will be transferred, and then it will be hybridized by using probe, and then final analysis by using auto radiography in this fourth step.